recording the class. The cardiovascular system is the 13th, I think, chapter of the Longman book. And in this chapter, we're going to talk about the heart formation and the vessels. Okay. At first, today we're going to talk about the heart itself, and from next session we're going to talk about the septums of the heart, and in the last session about vessels, heart vessels. We have studied the anatomy of the heart, and we know that the heart has 4 chapters, 2 atriums, right atrium and left atrium, the 2 chambers which are placed on the upper side of the heart, and right ventricle and left ventricle. Okay? The blood is coming into the right atrium and then right ventricle. We have the pulmonary trunk. After the pulmonary trunk, the blood is entering the lungs, and after exchanging the gases, it will return back into the left atrium and then into the left ventricle, and after the left ventricle we have the aorta, which send the blood to all parts of the body. Okay. Let's start the embryology of the heart. Almost in the middle of the 3rd week of the embryonic period, 3rd week, okay? At the middle of the 3rd week, at the time when the embryo cannot use the blood, and it is growing and cannot use the materials from other sides, or for example from diffusion, there would be a need for blood vessels to exchange the materials and give them beneficial materials to the embryo. Okay? At the middle of the 3rd week, the progenitor heart cells, the cells which want to make the heart, okay, are placed on the epiblast. The progenitor heart cells are placed on the epiblast adjacent to the primitive streak. Here we can see the epiblast of the embryo, and this is the primitive streak. These cells and also the progenitor heart cells are placed adjacent to the primitive streak. These cells are starting their migration from the primitive streak to go under this streak. Okay? These are starting their migration from the primitive streak to go under this streak. Also, we have some myoblasts and blood islands in the epiblast. Also, we can see some blood islands in the epiblast. The pointer is showing the myoblasts and the blood islands. The cells which have to make the upper part of the cells of the heart will migrate at first. The cells which have to make the outflow tract. The outflow tract of the heart will migrate at first, and then the cells which have to make the inflow part will migrate from the primitive streak. If I Is it better now? Okay. And what about noise? I don't know what to do. Okay. It's because of noise, is because of the volume. If I increase the volume, there would be some noise. Okay, I will explain. Okay, so I said the outflow tract cells will migrate at first, and then the cells which have to make the other parts of the heart, the inflow part, will migrate. The caudal part. At first the cranial part, the outflow, and then the cranial part, the inflow, will migrate. These cells are migrating into the cranial side. Here we have the primitive streak. The cells are migrating from this side and coming to the cranial side or cranial pole of the epiblast. Okay, like here. Look at here. This is the cardiogenic field. So they are migrating to the cranial pole of the heart, of the epiblast. And they are placing, look at here we have a membrane, buccopharyngeal membrane. They are placing in front of the buccopharyngeal membrane, buccopharyngeal or oropharyngeal membrane. These are placing at the anterior side of the buccopharyngeal membrane or oropharyngeal membrane. These cells are placed in the splanchnic layer of the mesoderm, lateral plate mesoderm. Here we can see the lateral plate mesoderm. Can you see? This is the ectoderm. The blue one is the ectoderm layer. This is the yellow one is the endodermic layer, and this is the lateral plate mesoderm. Okay, they are placing in the splanchnic layer of the lateral plate mesoderm. Here we can see them. The endoderm, which is placed under these cells, are inducing the cell of the splanchnic mesoderm to produce some blood islands or some myoblasts. The endoderm, which is placed under this part, that's notochord. The black one is the notochord for the ectoderm and the spinal cord. It is not related to our session, our chapter, okay? So, the endoderm is inducing the cells, the layer of the mesodermic layer, to produce the myoblast cells and also making some blood islands. Here we have an induction. So the blood islands will form. These blood islands are making some vessels with the way of vasculogenesis. These blood islands or these myoblasts are connecting to each other and making some vessels by or through vasculogenesis. These islands are connecting to each other, and at last they are forming a horseshoe-shaped tube. I will show you the tube. A horseshoe-shaped tube. The inside layer of this tube is the endothelium. It is called endothelium, which is surrounded by the myoblast. So these cells are connecting to each other and making some blood islands, the horseshoe shape, like the shoe of a horse. Okay, they are forming a horseshoe shape tube. The inside layer of the tube are the endothelium, is called endothelium, which is surrounded by the myoblasts. This place is called the cardiogenic region or field. This place is called the cardiogenic field. Here. Yes, the cardiogenic region or the cardiogenic field. The cavity which is around the cardiogenic field is the pericardial cavity. The cavity which is around the cardiogenic field is the pericardial cavity. So, the cells are migrating from the primitive streak and placing in the cranial region of the epiblast. This region is called the primary heart field. The endoderm is inducing the cells of the mesoderm also to make some blood islands. These blood islands are connecting to each other and making the horseshoe-shaped tube. The tube is surrounded by myoblasts, and this place is the cardiogenic field. The cavity which is around that is called the pericardial cavity. Okay. Also, we have a secondary heart field place. This place is in the splanchnic mesoderm, which is adjacent to the primary heart field. Look at here. This is the secondary heart field and this is the primary heart field. They are both adjacent to the primitive streak, primitive heart streak and field, and secondary heart field. Okay. Now here we can see the outflow tract and the venous pole or inflow tract. As you see, the outflow tract, I said that the cells which have to make the outflow tract are migrating at first, and then the cells which have to make the inflow part are making the inflow tract or the venous pole. This is the heart tube which is forming the horseshoe-shaped tube. And here is the secondary heart field. Here we can see the secondary heart field. Now, about the formation and the position of the heart tube. At first, the cardiogenic field is placed in the rostral region of the buccopharyngeal membrane or oropharyngeal membrane. Can you see that? At first, as we have seen here, the cardiogenic field is, this is the buccopharyngeal membrane. At first, we have the cardiogenic field in the rostral side of the buccopharyngeal or oropharyngeal membrane. But after that, when the neural tube is forming and making the brain vesicles, which cells? If you mean the cardiogenic cells, no. They are just migrating into the cranial side. Which one do you mean? Dorsally and laterally, no. The cells which are making the heart tube, no, I said they are migrating to the cranial side, to the rostral side compared to the oropharyngeal membrane. From the middle of the third week, the development of the heart is forming. From the middle of the third week. Okay? And it will continue until I will explain to you. Each part will happen when. Till, for example, 4 to 5th weeks. 4 to 5th weeks. Okay, let me continue. We said that these cells are placed on the rostral side of the oropharyngeal membrane. By the development of the neural tube and the vesicles of the brain, these ectoderm will growing. Look at here. The ectoderm will grow, and the speed of its growing is very much, mostly at the cranial side. The speed of the brain growing is very much, mostly at the cranial side. And the brain vesicles are forming like this. By the formation of the brain vesicles, I think slide number 4. Slide number 4. Okay. By the growing of the neural tube, the brain vesicles are placing on the cardiogenic place. They are placing on the cardiogenic place and moving the cardiogenic field into the near to the neck. Okay, they are growing on the cardiogenic field and the precardial cavity, and they are moving the cardiogenic field into the neck and the thoracic cavity, like these pictures. Pay attention, please, to these pictures. So, the cranial nervous system is growing into the cranial side rapidly and extend over the central cardiogenic region and the future pericardial cavity. And also we have some cephalic folding or cranial folding of the embryo. Okay? We have some cephalic or cranial folding of the embryo. Look at here. The cranial and the caudal sides of the embryo are folding. So because of this folding also, the cardiogenic field is moving into the neck and the thoracic place, to the cervical region. It would be better to say the heart is moved at first to the cervical region and finally to the thorax. As the embryo folds cranially, it is also folding laterally. Look at here, before the folding, I want to explain about the oropharyngeal membrane. In this slide, we can see that the oropharyngeal membrane will place on the rostral side of the cardiogenic field. At first, the cardiogenical cells or myoblasts migrate on the rostral side of the oropharyngeal membrane. Then, by the cranial folding, the heart will place in the cervical region. So the oropharyngeal membrane is in the anterior side compared to the heart tube or the cardiogenic field. As we said, we have some lateral folding beside the craniocaudal foldings in the embryo. Here we can see the craniocaudal foldings. And in this slide, the next slide, we can see the lateral foldings of the embryo. These are the lateral foldings. The lateral foldings will connect the blood islands and the angiogenic cells to each other. Look at here. At first, so we have two endocardial tubes on each side, one in the right and one in the left side. We have two cardiogenic tubes, and then by the lateral folding of the embryo, these two tubes are connecting to each other and forming one endocardial tube. The inner layer of this tube are the endothelium, which is surrounded by the myoblast, which is called myocardium. As I said, the central part of this tube, which is called the horseshoe-shaped tube, expands to form future outflow tract or the ventricular region, which I explain in the next slides. Did you get these foldings? How they occur? We have two foldings in the embryo. One, the craniocaudal. If I can find, yes, I will send you. You can search it on the internet yourself. We have the craniocaudal foldings. The craniocaudal foldings are making the heart to move into the cervical region and also the thorax. And the lateral folding of the embryo are connecting the angiogenic cell clusters to each other and making the endocardial tube. The endocardial tube, which is a horseshoe-shaped tube, has the endothelium in its inside part, and it is surrounded by the myocardium. It is surrounded by the myocardium. Now you have to listen to my explanation. Then after the class you can search. And if I find something, I will send it to you. Now just listen, okay? So here we have the endocardial tube. As we can see, there is a layer of mesocardium which is connecting the endocardial tube to the dorsal wall of the body. As we can see, there would be a dorsal mesocardium which is connecting the endocardial tube into the dorsal wall of the body. This is called dorsal mesocardium. So the heart tube is connected to the dorsal wall by the dorsal mesocardium, but yes, the dorsal side of the pericardial cavity. But there is no ventral mesocardium. There is no ventral mesocardium. Look at here. Let me show you the dorsal. Yeah. Here we can see the heart tube, the inflow and outflow side, the heart tube, and this is the dorsal mesocardium. At first, the dorsal mesocardium is a kind of a plate, and it is present in all sides. But after some time, there would be some holes in the mesocardium, in the dorsal mesocardium. The dorsal mesocardium is derived from the secondary heart field. Okay, the dorsal mesocardium is derived from the secondary heart field. So, the middle section of the dorsal mesocardium disappears. As we can see here, the middle side is disappearing. So, the heart is just connected to the dorsal wall in the inflow and outflow sides. The heart is just placed, connects in both sides to the precardial cavity. The hole which occur and appear in the dorsal mesocardium is called the transverse sinus of the pericardium. Do you remember the transverse sinus? We had two sinuses in the pericardium, oblique and transverse. This is the transverse sinus of pericardium. So the heart tube will suspend in the pericardial cavity from these two sides, and between them we have the transverse pericardial sinus. Yes. After the folding of the heart tube, we can see the last form of the transverse sinus. During these events, the myocardium thickens and secretes a layer of extracellular matrix which is rich in hyaluronic acid. The myocardium thickens here. The myocardium is secreting a hyaluronic acid layer, which is called the cardiac jelly. This jelly separates the myocardium from the endocardium. This jelly separates the myocardium from the endocardium. Look at here, we have seen the cardiac jelly. Hyaluronic acid, yes. This hyaluronic acid layer is called cardiac jelly. It is separating the endocardium and the myocardium. Its function, it would be like a layer of the heart. Okay. Also, we can see some proepicardial organs on the mesenchymal cells which are located at the caudal border of the dorsal mesocardium. At the dorsal side of the mesocardium, we can see some proepicardial. Look at here, also here we have the cardiac jelly, endocardial tube, and this is myocardium. It's before the folding, and after that these tubes are connecting to each other and we will see just one cardiac tube. So we said that near this part, at the caudal part of the dorsal mesocardium, we can see some proepicardial cells. These proepicardial cells are migrating on the heart tube and forming this. They are proliferating and migrating on the myocardium and forming the epicardial layer. Forming the epicardial layer, which is called the epicardium of the heart. So now, in this part, the heart is formed from, okay, I will explain. 3 layers. Look at here, we can see endocardium and myocardium. The myocardial cells are secreting a hyaluronic acid layer, which is called cardiac jelly. The cardiac jelly is placed between the myocardium and endothelium. Myocardium and endothelium. Also, at the caudal side of the dorsal mesocardium, there would be some proepicardial cells or region. Proepicardia. The proepicardial cells also migrating on the heart tube and proliferating and forming the epicardium. Okay? After that, the cardiac tube or the heart tube has 3 layers: endocardium, myocardium, and epicardium. Endocardium forming the internal lining of the heart. Myocardium is the muscular wall of the heart, and epicardium is also called the visceral pericardium. The outer layer, the epicardium, is responsible for the formation of the coronary arteries. It's so important. The proepicardial cells from the caudal side of the dorsal mesocardium are proliferating and forming epicardium. These epicardial cells are also responsible for forming the coronary vessels on the outer side of the heart. Okay? Understood? Now about the formation of the cardiac loop. Any question till this side? Proepicardial cells. Myocardium what? What is the question, myocardium? Please ask complete question. At this stage, the heart has come to its it's just in the cervical region. The heart is in the cervical region, and after that it will place in the thorax. Now the heart is in the cervical region. Like this slide. Look at here. Like this. Can you see? It's the cervical region. I didn't talk about the neural crest cell. Why are you asking? Let me continue, please. Please exit the class and enter again if you can see the slides. Refresh. Now about the endocardial tube formation. The heart tube how it moves and changes location after completing its development to somehow. I said by the embryonic folding, craniocaudal and lateral folding, the heart is moving into its final destination. At first in the cervical region, look at here, at the time that the heart is forming, the other organs of the embryo also are forming. For example, the brain vesicles, okay, the digestive system. All of these happenings are occurring with each other. So they are affecting each other and making the heart to place into the last destination, to its last destination. Okay. Now about the formation of the cardiac loop. This is the epicardial cavity, precardial cavity, and this is the heart tube. Listen please. This is the heart tube. We want to talk about the formation of the cardiac loop. The heart tube continue to elongate till the 23rd day. Till the 23rd day. At this time, it is starting its looping. The cardiac tube is starting its looping. How? Look at this picture, let me explain. At first, just some parts of the cardiac tube are placed in the pericardial cavity. Let me show you again this. Look at here. Here we have the primitive precardial cavity, and before folding, just some parts of the cardiac tube are placed in the pericardial cavity. But after folding, all parts of the heart have to enter into the precardial cavity, like the final form. In this slide also we can see the dorsal mesocardium and the folding, which forms the transverse sinus formation. And here we can see the forming. This part is showing the precardial cavity. At the end, we can see that all parts of the tube would place into the pericardial cavity. But how? What are the formation? The cephalic portion of the tube bends ventrally. This is the cephalic portion, okay? This is the cephalic portion of the heart tube. Listen, this part is so important. This part is bending ventrally, caudally, and to the right. Ventrally, caudally, and to the right side. But the atrial or the caudal portion bends the atrial or the caudal portion, this part. The caudal portion bends or shifts dorsocranially and to the left. Dorsally, cranially, and to the left. Yes, opposite. So, this part bends ventrally, caudally, and to the right. The atrial portion bends dorsocranially, look at here, dorsally, cranially, and to the left. The caudal portion, the cranial portion or the cephalic portion of the heart tube is forming some parts like the bulbus cordis and the ventricle. But the caudal part of the heart tube forms the atrium and the sinus venosus. The caudal portion forms the atriums and the sinus venosus of the heart. By this looping, the atrial portion is moving inside the precardial cavity. You can see in this slide. It is moving inside the precardial cavity. These happenings will change because of the changes in the mitosis, or for example forms or the shapes of the cells. The cells will proliferate in some parts more, for example in these parts, and the cardiac looping is formed. The cardiac looping is complete by day 28. So it starts from day 23 and end by day 28. While the cardiac loop is forming, local expansions become visible throughout the length of the tube. We can see that some parts of the tube are expanding. It's clear in this part. Look at here, for example, the ventricle side with the bulbus cordis. Some parts are expanding. The atrial portion or the atrial part in the caudal side of the heart tube initially a paired structure outside the pericardial cavity. It forms a common atrium, and after that it enters the precardial cavity. After that it enters the pericardial cavity. Look at here, we have atrioventricular junction. This is the atrium, the common atrium, not here we don't have atriums, okay? Just a common atrium. And here we have atrioventricular junction between the red and the blue side. The atrioventricular junction remains a narrow part and form atrioventricular canal. So it remains a narrow part and forms atrioventricular part. It connects the common atrium to the early embryonic ventricle. The blue part is the embryonic ventricle and this is the atrium. The atrioventricular canal is connecting the atrium to the primitive ventricle. What about bulbus cordis? The bulbus cordis has three parts. Yes, the blue is ventricle. The red one is the atrium, the blue is the ventricle. Okay? It has written, look at here, the blue one is ventricle and this is the atrium. The bulbus cordis has three parts, white, black and green. What happened to these three parts? It is also narrow except for its proximal third part. It is also narrow, as we can see, except in the blue part. Except in this green part, sorry. These parts are narrow and this is not. The portion, the green portion, will form the trabeculated part of the right ventricle. The proximal third of the bulbus cordis, the green part, will form the trabeculated part. No, this is, look at here, let me explain to you. This is the inflow part of the heart tube. Okay, the red part or the sinus venosus is the inflow. So we call this part the proximal and distal. So the white portion is the distal part, the outflow part, okay? No, I didn't talk about the right atrium. Just listen, I will explain our parts. I said that here we have a common atrium. Common atrium means just one atrium. Yes, auricle. Then we have the blue one, which is the ventricle, and the bulbus cordis has 3 parts. Yes, the green one is the proximal part, proximal third. Then we have the black, and at the distal side we have the white part. The green part is forming the trabeculated part of the right ventricle. So we can see that the blue one is the left ventricle, the blue portion. The general sentence for that is the primitive ventricle in the heart tube is forming the left ventricle. Okay? The primitive ventricle is forming the left ventricle, and the proximal third of the bulbus cordis is forming the right ventricle, the trabeculated part of the right ventricle. Any question? Please don't ask about the attendance link. Just listen to the explanations. They are too hard. The blue part is the primitive ventricle. The primitive ventricle is forming the trabeculated part of the left ventricle. And the green part is the proximal third of bulbus cordis, which is forming the trabeculated part of the right ventricle. Any question? Then, the mid portion of the bulbus cordis is called the conus cordis. The mid portion, or the black one. No. Blue, no. White, black, and green are parts of bulbus cordis. These 3, white, black, and green, are parts of bulbus cordis. The mid part, or the black one, is called the conus cordis. Conus cordis will form the outflow tracts of both ventricles. Conus cordis is the black one. All of these three are called bulbus cordis. Conus cordis is just the black one. It's like a cone. The black one is the conus cordis, the mid portion of the bulbus cordis, and is forming the outflow tracts of both ventricles, right and left ventricle. The outflow tracts of both ventricles. The distal part, I mean the white part of the bulbus cordis, is called truncus arteriosus. The white part is called truncus arteriosus. The truncus arteriosus will form the root and proximal portion of the aorta and pulmonary artery. The distal portion of bulbus cordis is called truncus arteriosus, is forming the root of aorta and pulmonary trunk. The black one is conus cordis. The white one is truncus arteriosus. Listen, I will explain one more time, okay? We have the atrial side in red. Just listen. Don't ask any question, just listen. We have a part in red which is the atrium portion and the sinus venosus. Atrium and sinus venosus. It is placed on the caudal side of the heart tube, and this is the inflow portion. Okay? After it is forming the common atrium, just one atrium. At this level, we have just one atrium, and this is the red one. After that, we have the blue part. It's the primitive ventricle and forming the trabeculated portion of the left ventricle. The blue part, the primitive ventricle, is forming the trabeculated part of the left ventricle. And the other part from this line to the end is called bulbus cordis. Bulbus cordis has three portions itself: green, black, and white. So, bulbus cordis has three portions itself: green, black, and white. The green part is forming the trabeculated part of the right ventricle. The black part of bulbus cordis is called conus cordis. And the conus cordis is forming the outflow of both ventricles, right and left. The outflow or the smooth part of both ventricles, right and left ventricles. The white part is called the truncus arteriosus and is forming the root of aorta and pulmonary trunk. Okay? You're welcome. Very good. The others. Do you have any question? Do you have any question? Okay, let me continue. I said that there would be a junction between the atrium and the blue part, I mean the left ventricle. We call that the atrioventricular canal. Okay, we have. The next time you are asking me about the attendance link, I will give you a negative point for your midterm. Everybody. So, we have the atrioventricular canal at this part, between the atrium and the ventricle. Also, we have another junction between the ventricle and the bulbus cordis. Between the ventricle and bulbus, because I'm explaining the lessons and all the time you are asking for the attendance link. Okay, another junction is between the ventricle and the bulbus cordis. No problem. This junction between these two parts externally indicates by the bulboventricular sulcus. It also remains narrow. Bulboventricular sulcus. Yes, and it's also narrow. We get here bulboventricular sulcus. It is called the primary interventricular foramen. The second junction is between the ventricle and the bulbus cordis. This part, between the green and the blue colors. It remains also narrow and is called bulboventricular sulcus. This is the place of the primary interventricular foramen, because we said that the blue part is the left ventricle and the green part is the right ventricle, okay? So, the junction between these two parts is called primary interventricular foramen, between two ventricles. So after these events, we can see that the cardiac tube is near to its final form and entered into the primitive precardial cavity. When the looping is completed, the smooth-walled heart tube begins for primitive trabeculate in two sharply defined areas, just proximal and distal to this interventricular foramen. I have explained this part in the anatomy classes. Look at here, this is the ventricle, okay? Do you remember that in the right ventricle we said it has two parts, inflow and outflow? Do you remember? Okay, the inflow part of the right ventricle was trabeculated. It has papillary muscle, bridge-like muscle, ridge-like muscles. This is the trabeculated part, and the outflow part of the right ventricle had a smooth wall. The outflow part of the right and left ventricles had smooth wall. Yes, for the inflow of the blood. Yes, very good. The trabeculate cornea of ventricle for inflow of the blood. No, not the valves. Don't confuse. No. I did not talk about the tricuspid or mitral valves. I'm just talking about two parts of the ventricles, inflow and outflow. Okay? The inflow part of each ventricle, listen, the inflow part of each ventricle has trabeculated muscles and it is a row-walled part. The outflow part has a smooth wall part. So at this time, we can see that the muscles at both sides of the interventricular foramen are starting to trabeculate and forming the trabeculate part of the right and left ventricles. They are just proximal and distal to the primitive interventricular foramen. This is the interventricular foramen, and just proximal and distal to this foramen we can see the trabeculated part of the left ventricle and the right ventricle. So, the primitive ventricle, I mean the blue color in that slide here, the primitive ventricle is forming the trabeculated part of the left ventricle, and the green part of bulbus cordis is forming the trabeculated part of the right ventricle, these two parts. In that slide, for example, you can say that this is the blue color and this part is the green color. Okay? It's the blue color and this is the green color from the previous slide. The conotruncal portion of the heart tube, I mean the conus arteriosus, the conus cordis. The conus cordis here, the black part. The conus cordis portion of the heart at first is placed on the right side of precardial cavity. Look at here, it's the conus cordis. At first, it is placed on the right side of the precardial cavity. But it gradually shifts to more medial position. It shifts to medial position. Look at here, it is shifting and coming into the medial position in the precardial cavity. This change in position is the result of the formation of two transverse dilation of the common atrium. You have seen that, look at here, the common atrium, this red part, is entering into the precardial cavity and placed behind the blue, green, and black parts. Here, we can see this is the primitive common atrium. This atrium is making two dilations, two expansions in both sides. These dilations are changing the position of the conus cordis in the midline of pericardial cavity. Okay? So, the last position of the conus cordis is in the middle part of the pericardial cavity because of the two dilations in the pericardial, in the primitive common atrium. Because the primitive common atrium wants to form the primitive right atrium and primitive left atriums. Clear? Okay. Very good. This slide is also showing the rotation of the heart tube. It is the ventricle, and this is the atrium part. In this part also you can see the looping of the heart. Here also we can see the looping. This is the outflow tract. This is the atrium part and sinus venosus, which coming into the pericardial cavity. This is the primitive atrium, primitive left ventricle. I don't know which slide is it. Let me check. Let me check and tell you. I'm slide number 14. I'm talking about slide number 14. Okay. Also in this slide, I just found some pictures of the cardiac looping and for you to understand them better. Okay, let's have a break for 5 minutes. And no, we have 3 sessions, 1/3. And please click on the attendance link. I will put you for this part. Mr. Fassi will share it in your other groups. I can't. Mr. Fassi, if you're present, could you please put it into your WhatsApp group? Is Mr. Fassi present or not? Oh, thank you, Mr. Muhammad. Your family is Afraish? If I spell it right. Your name is Afraish. Your family is Muhammad, yes? They are both named? Okay. And you're a boy, yes? Very good. You are a clever student. You're welcome. Okay, share the link because I think Mr. Fassi is absent. And you have 5 minutes break. Then I will return. Okay, I have closed the link because I gave 98 responses. Take a break and then I will. Okay, are you ready? About the blood circulation after the completion of the looping, we have to say that the blood will enter to the primitive atriums from the sinus venosus part, and after that it will enter to the left ventricle, passing through the primitive interventricular foramen, entering the primary right ventricle, and then by passing through the conus cordis, it will enter to the truncus arteriosus, the aorta and pulmonary trunk, the aortic arcs at this stage. I will explain the blood circulation completely in the last session, okay? But now, let's talk about some of the laterality effects on the heart development. Some of the genes like FGF8 and nodal genes, expression of these genes in the left side of the embryonic disc will have some important effects in the laterality of the heart development. What does it mean, the laterality? It means the looping and bending of the heart tube. And also we have PTX2. Also we have PTX2 at the left side, which is the results in the polarity of the heart-forming cells. It means that if these genes do not express or express in the right side, something will happen and the heart looping will change in vice versa direction. Serotonin or 5-HT in the left side initiates the laterality sequence. Look at here, the 5-HT or serotonin is also initiates the laterality sequence. These changes take place on days 16 to 18th of development. 16 to 18th of development. If something happened to these genes, or also the looping, or for example they express in the right side or anything, there would be some problems. We call them the laterality problems. These abnormalities are including dextrocardia. For example, dextrocardia will occur in some people who these genes are expressing in the right side, and instead of left looping of the heart tube will loop in the right side, so their heart will place in the right side of their chest. We call that dextrocardia. Okay? The heart will place in the right side of the chest. This defect may be induced during the gastrulation, but dextrocardia may occur with situs inversus. Situs inversus is a condition where all the organs will occur in vice versa sides. Reversal sides, okay? In dextrocardia, just the heart is placed in the right side of the chest, but in situs inversus, all of the organs also are in the reversal position. Also, dextrocardia may occur with some laterality sequences which are called the heterotoxy. Yes, it's right. Dextrocardia is a condition where the heart is placed in the right side of the chest. Dextrocardia. If all of the organs beside the heart placed in the reversal position, we call that condition, the situs inversus. Situs inversus. Situs. Situs inversus. Yes, in situs inversus we can see the reversal position of all of the organs. Okay? But there would be some other conditions with the name of heterotaxia. What will happen? In heterotaxia, only some organ positions are reversed. In heterotaxia, only the position of some of the organs will be reversed. We have two kinds of heterotaxia. One of them is right-sided bilaterality and the other is left-sided bilaterality. What does it mean? Right-sided, let me write it for you. Right-sided bilaterality and left-sided bilaterality. In this kind of conditions, only some organs are reversed. The position of some organs are reversed. In right-sided bilaterality, these kind of people, the liver, no, the spleen. The physician will understand this situation by the form of the spleen and the number of the spleen. In right-sided bilaterality, they have aspleny or hypoplastic spleen. But in left-sided bilaterality, they have polyspleny. They have more than one spleen. Yes, the position of the stomach also changes. So listen, the laterality condition have many different forms. In dextrocardia, there would be just the reversal of the position of the heart. In situs inversus, all of the organs have a reversal position. Dextrocardia and situs inversus person do not have any other clinical problems. They are okay. But the other form are the bilaterality. Yes, it's rare. Situs inversus, I think, is 1 in 7,000. 1 in 7,000, I think. So, the bilaterality effect is when it is right. Yes, it's genetically because of the genes nodal, serotonin, LEFT2, and these genes. So, if it is the right-sided bilaterality, they will suffer hypoplastic spleen or aspleny. They don't have spleen. And if it is left-sided, they have polyspleny. They have more than one spleen. They have more than one spleen, okay? For example, this is an abnormality related to mistake folding. Okay, now let's talk about the development of the sinus venosus. You have seen that the proximal portion of the heart tube is called, yes, they perform spleen function, but they are small. About the development of the sinus venosus. Listen, we have seen that the proximal portion of the heart tube is called sinus venosus. The sinus venosus in the middle of the fourth week receives venous blood from right and left sinus horns. On the middle of the fourth week, it receives, let me show you the sinus venosus again. Okay. Look at here, this is the sinus venosus, this one. It was attached to the atrium, common atrium. Here. Now, at the middle of the fourth week, it receives the venous blood from the right and left sinus horns. Each horn receives blood from 3 important veins. One of these veins is vitelline or the omphalomesenteric vein. Vitelline on each side. Vitelline or omphalomesenteric vein. The other vein is the umbilical vein. This one. And the third one is the common cardinal vein. Common cardinal vein receives the anterior cardinal and posterior cardinal veins. At the beginning of the embryonic period, the connection or the communication between the sinus venosus and the common atrium is very big. Can you see that? The sinoatrial junction is very big at first. But the entrance of the sinus will then shift to the right side just. It will shift to the right side. But why? Why this will happen? It is caused by the left to right shunts of the blood. It is caused by the left to right shunts of the blood. It means the blood flow is shunting from the right side to the left side. And from the left side, to the right side. We call that left to right shunt. Left to right shunt. What will happen? Some of these veins, for example, umbilical and vitelline, and these veins will obliterate. Some of these veins will, shunt, obliterate. Which ones? One of these veins which will obliterate is the right umbilical vein. Look at here. The right umbilical vein is obliterated, and also the left vitelline vein during the 5th week. The left vitelline vein. After some time, the left sinus horn rapidly loses its importance. This horn, the left horn, will rapidly loses its importance by the obliteration of its veins. The left common cardinal vein is also obliterated at week 10. The left common cardinal vein is also obliterate on the 10th week. And the thing that just remains from the left sinus horn is the oblique vein of the left atrium. It would be just, look at here, it would be just the oblique vein of the left here, of the left atrium. No. Okay? The oblique vein of the left atrium and also the coronary sinus are the remains part of the left venous horn. So, the remaining parts of the left venous horn are the oblique vein of the left atrium and the coronary sinus. The right sinus horn and veins are also enlarged greatly. Can you see? Look at here. They are making enlargement on the right side. The right horn formed the only communication between the original sinus venosus and the atrium. Now we can see the sinoatrial junction is just between the right horn and the atrium, not the left side. This part is attributing and incorporating the smooth valve part of the right atrium. This part, I mean the right venous sinus, is forming the right portion of the right atrium. The entrance of the sinus venosus into the atrium is called sinoatrial orifice. Look at here. This is the right atrium and this is the left atrium. The entrance of the synovial venosus into the right atrium is called the sinoatrial orifice. It has two valves. Sinoatrial orifice has two valves. Right and left venous valves. Right and left venous valves. These valves are dorsocranially forming a ridge known as the septum sporium. In the dorsocranial side, these valves are connected to the wall of the right atrium with a septum which is called the septum sporium. Here. By entering the sinus venosus into the right atrium, this part will enlarge and, as I said, will make the smooth valve part of the primitive atrium, the right atrium. The right sinus horn is incorporated into the wall of the atrium, and the left valve and septum sporium are moving into the left side. Look at here. These valve and septum sporium are moving into the left side of the right atrium. The superior portion, the superior part of the right venous valve disappears entirely. By attaching these two parts into the interatrial septum, the superior portion of the sinus will disappear entirely, but the inferior part will develop into two parts: a valve for inferior vena cava and a valve for coronary sinus. The valve for inferior vena cava and a valve for coronary sinus. The crista terminalis will show the part will separate the smooth wall part of the right atrium and the raw part of the right atrium. So the origin of the smooth wall part of the right atrium is the sinus venarum, and the origin of the raw walled part of the right atrium or the auricle is the primitive atrium. Okay, clear? Any question? Yes, the smooth walled part is formed from the sinus venarum. The raw walled part is from the primitive atrium. If you have any question, you can ask, and if not, this session is finished. You're welcome.